Welcome to MTOZ Biolabs channel. Today, we will provide an overview of untargeted metabolomics. In living systems, thousands of small molecules, known as metabolites, are constantly synthesized, broken down, and recycled. These metabolites act as the chemical signals that keep cells running, offering a sensitive reflection of physiological and pathological changes in the body. Metabolomics is the comprehensive qualitative and quantitative profiling of small molecule metabolites in biological systems and their condition-dependent changes to link metabolic states with pathways, phenotypes, and physiological functions. Because metabolites occupy a central position between genes and phenotypes, metabolomics is recognized as the bridge that connects molecular mechanisms with biological functions. When we talk about metabolomics, there are actually two main approaches: targeted and untargeted. Targeted metabolomics focuses on a limited set of predefined, well-characterized molecules, measuring them with high precision. Untargeted metabolomics does not rely on predefined hypotheses. Instead, it comprehensively profiles the full range of metabolites in a sample. In this way, we not only capture known compounds but also uncover previously uncharacterized metabolites. Untargeted metabolomics is a discovery-driven tool that helps us find novel biomarkers, uncover hidden metabolic pathways, and open doors to new biological insights. And with today's high-resolution mass spectrometry and big data analytics, we can dive deeper into the metabolic network than ever before. The experimental workflow of untargeted metabolomics starts with sample preparation. Samples such as blood, urine, tissues, or cells must be collected and stored under standardized conditions to ensure metabolite stability. Next comes metabolite extraction and purification, where proteins and other large molecular impurities are removed, leaving the small molecule metabolites ready for analysis. In certain cases, we perform derivatization. For example, in GC-MS analysis, derivatization improves the stability and detection sensitivity of specific metabolites. The workflow then moves to its core step: chromatographic separation combined with high-resolution mass spectrometry detection. Using LC, GC, or UHPLC greatly enhances the separation of complex mixtures, while high-resolution MS together with the data-independent acquisition mode provides comprehensive coverage and precise detection. After generating the raw data from mass spectrometry, we move into the data analysis stage. The first step is data preprocessing, which includes noise reduction, retention time correction, peak detection, integration and chromatogram alignment, and finally data normalization. These steps ensure accuracy and consistency across all samples. Next, we perform experimental quality evaluation, checking data reliability before moving forward. The core of the analysis includes several layers. In metabolite identification, we summarize the number of identified metabolites and classify them into chemical categories. Then we conduct intergroup differential analysis, combining univariate and multivariate statistics to identify differential metabolites between groups. Finally, we carry out bioinformatics analysis of differential metabolites. This involves clustering and correlation studies, as well as KEGG pathway annotation and analysis, to reveal the underlying biological significance. These steps transform raw data into meaningful biological insights. Once data analysis is completed, the results are presented using multiple visualizations to highlight different aspects of metabolomic study. Here, the PCA plot shows the overall distribution of samples and reveals natural clustering trends without prior classification. The PLSDA plot, in contrast, is a supervised method that emphasizes separation between predefined sample groups. The volcano plot highlights metabolites that are significantly upregulated or downregulated, combining fold change with statistical significance for rapid screening. As shown in the figure, each dot corresponds to a metabolite. Red dots represent significantly upregulated metabolites, and blue dots represent significantly downregulated ones, while gray dots represent non-significant differential expression metabolites. The heat map provides an overview of metabolite abundance across all samples, with clustering to reveal group-specific expression patterns. In this heat map, each column represents a sample, and each metabolite is visualized in a row. Red indicates a high abundance, and blue indicates a low abundance of metabolites. The KEGG enrichment scatter plot provides an overview of significantly enriched metabolic pathways, helping to link differential metabolites to biological functions. Each dot corresponds to a KEGG pathway in the plot. The dot size reflects the number of differentially expressed metabolites in the pathway. Larger dots indicate more DMs. The color of the dot represents the p-value. Smaller p-values indicate greater statistical significance. Finally, a metabolite network diagram maps the differential metabolites onto a curated metabolic network, illustrating their interconnections. In the diagram, each node represents a metabolite, while the edges indicate biochemical reactions or associations between them. Together, these visualizations provide a comprehensive and intuitive interpretation of metabolomics data. Untargeted metabolomics is not just about data and plots. Its findings translate into real biological and clinical applications. Untargeted metabolomics enables the discovery of clinically relevant biomarkers by comparing metabolic profiles between healthy and diseased states. Such comparisons also shed light on disease mechanisms, revealing pathway-level alterations that link metabolic dysregulation to progression, and pointing to potential sites of intervention. These insights naturally extend to targeted therapy development, where metabolic mapping highlights treatment-responsive pathways and metabolites that can serve as therapeutic targets. At the same time, drug metabolism studies benefit from this approach by tracking pharmaceutical biotransformation, uncovering unexpected metabolites, and clarifying off-target effects to improve pharmacokinetics and safety evaluation. Moving beyond medicine, comprehensive profiling monitors metabolic responses to diet and lifestyle, supporting insights into nutrient utilization, functional food effects, and personalized nutrition. Despite its broad potential, untargeted metabolomics faces several challenges. First, metabolites are highly diverse, including carbohydrates, lipids, amino acids, nucleotides, and other small molecules, with greatly varied chemical properties. Metabolites also show rapid dynamics. Their concentrations can shift instantly in response to environment, diet, or disease states, making them difficult to capture consistently. The concentration range is vast, as abundant and trace-level metabolites coexist in the same sample, complicating the detection of low-abundance compounds. Structural diversity adds further difficulty, since isomers and close analogs are common and hard to distinguish accurately. Finally, many metabolites are still unknown. Existing spectral libraries provide only partial coverage, and accurate identification depends heavily on available references. MTOZ Biolabs provides end-to-end untargeted metabolomic services designed to overcome the key challenges in this field. Our advanced LC-MS/MS and GC-MS platforms provide broad metabolome coverage, spanning carbohydrates, lipids, amino acids, nucleotides, and beyond. Time-resolved sampling and optimized preservation protocols capture rapid metabolic fluctuations influenced by environment, diet, or disease. High-resolution instruments with extended dynamic range, combined with optimized sample preparation, allow reliable detection of both abundant and trace metabolites. Structural complexity, such as isomers and analogs, is addressed through multidimensional separation and expanded spectral libraries, enhancing identification accuracy. Meanwhile, in-house curated databases integrated with KEGG and HMDB, together with AI-driven bioinformatics platform, strengthen metabolite identification and annotation. By integrating state-of-the-art instrumentation, robust bioinformatics, and rigorous quality management, MTOZ Biolabs empowers researchers to fully exploit the potential of untargeted metabolomics for biomarker discovery, disease mechanism studies, and translational applications. Thank you for watching and feel free to contact us for more information.